Alright, hi guys. Let's walk through this Mendelian genetics worksheet. So we'll start with this one first. Go ahead and put your name at the top. Alright, so this one starts off with the question, fur color in hamsters is controlled by one gene with two alleles. Okay, so here's our chromosomes. That means we got one gene and there's two alleles. The alleles are expressed with incomplete dominance. So incomplete dominance means if the hamster has the genotype big B, it's going to have black fur. If it has big B, it's going to have gray fur. And if it has little b, it will have white fur. And so that's what we know about the hamster, since it's incomplete dominance. Okay. Let's see. So a heterozygous hamster has offspring with another heterozygous hamster. Well, what's heterozygous mean? That means you have one of each allele. So we have one hamster that is, we have one hamster that's big B, little b. We have another hamster that's big B, little b. And what we need to figure out is what proportion or percentage of their offspring will be homozygous recessive. So what percentage is going to be little b, little b? Well, we take each parent and we split their alleles and we put them up here. And so what that basically says is, okay, this parent has the big B on one chromosome, they have the little b on the other chromosome, just like up here. So that means that each hamster's genotype looks like this, and they're only going to be able to pass on one set of chromosomes, so they're either only going to be able to pass on the big B or they'll be able to pass on the little b, and so that's why we split them into their own columns. And we'll do the same thing for the other hamster because they have the same genotype. And then all you do is just create the matrix. So 1 out of 4 is going to be homozygous dominant. 2 out of 4 will be heterozygous, right? So we trace the big B down, little b across, little b down, big B across. And then finally this last one, what percentage will be homozygous recessive? Well, that'll be 1 out of 4. So we could say 1 out of 4 or 25%, either way. Or you could do 0.25. All of those are exactly the same thing. Okay? All right, so next question. Eye color in chimpanzees is coded for by one gene with two alleles, so big A, little a. Big A is dominant and codes for dark eyes, so it's complete dominance. Little a is recessive and codes for light brown eyes. So I give you two different chimpanzees and I give you their phenotype. So I say a black-eyed chimpanzee has offspring with a light brown-eyed chimpanzee. Half their offspring are black-eyed and the other halves are light brown-eyed. So what are their genotypes? Okay, so we know that parent A is either genotype capital A, little a, or capital A, right? Parent B, we know because they have light brown eyes, they have to be homozygous recessive. So we know that their genotype is little a, little a. So I'm going to take parent A. I'm going to take parent B. Parent B's genome looks like this. He's got the gene for eye color, and he's got 2 copies of the little a. So one of these chromosomes gets put over here, so that's the little a. The other one gets put over here in the other row. So that's one parent, right? Now the other parent could be either capital A, little a, or big A, big A. So let's think about this, right? If half the offspring have black eyes and half have light brown eyes, well, that means half the offspring are going to have the genotype little a, little a. The other half of the offspring, well, we don't know, right? So the only way that this offspring can be created is if the other parent, parent A, also has a little a. So we'll put the other parent's possible genotype up here and we'll see if it makes sense. Okay, so the other parent, parent A, who has dark brown eyes or dark black eyes, has the genotype big A, little a. That means that half of his offspring will carry his big A. Half of the offspring are going to carry the big A, and half of the offspring are going to get his little a. Because the other parent is homozygous recessive, it's always going to pass on the little a. So every offspring is going to have at least one little a. And because half of them have the homozygous recessive phenotype, we know that they're getting another little a from one of the parents, parent A. And since half have the black eyes and half have light brown, we know that big A is in there too, and so that's what tells us what the parents' genotypes are. So the parents' genotypes are big A, little a, and little a, little a. Cool. All right. So the genotype would be, big A, little a, and little a, little a. Those are the two genotypes. Okay. The previous question was a certain type of breeding experiment used to determine the genotype of a dominant phenotype. This is example of a, done this before. We did this with hamsters in the lesson. I'm actually not even going to tell you the answer to that. You're going to have to look it up. Okay. So you should be able to find that one real easy, or you should know it already if you've already watched the video, as long as you're not doing things out of order. Okay. Next question. Pea plants have 2 genes, each expressed with complete dominance, and 2 alleles for each gene. So what does that mean? That means they have 2 chromosomes. That probably looks something like that. That means on top of the chromosomes they got 1 gene, and on the bottom of the chromosome they got another gene. Okay. And it looks like the 2 genes are going to be Y and R. Okay. Now, what it says is it says a heterozygous plant for both traits is crossed with a double homozygous recessive plant. Well, let's figure this out right now. Okay. A heterozygous parent, what is that even going to look like? Well, if this gene up here is the Y gene and they're heterozygous, that means they've got a capital Y and they've got a lowercase y. Right? If they're heterozygous for both traits, that means they have a capital R and a lowercase r. So that's what the genome of the one parent looks like for both traits, right? And we cross this individual with an individual who is homozygous recessive for both traits. So that means we cross them with this individual. And this individual is going to have a lowercase y, a lowercase y, a lowercase r, and a lowercase r. And so what we need to ask is we need to say, okay, it's basically saying if 100 plants are, if we cross these 2 individuals together and then they make 100 babies, right? It said how many will we expect to be yellow and round? Well, if capital Y codes for a yellow pea and capital R codes for a round pea, basically we just need to know how many peas are going to have one dominant allele from each gene in those individuals. So how do we figure this out? Well, the way I set up this table is I set up this table to say, okay, what are all of the possible gametes, sperm and egg, that the double heterozygote individual could make? Well, we know that they could pass on a big Y and a little r. So let's put that right here. Big Y and a little r. Okay, that's one possible combination, right? Because through meiosis, through recombination, this little r can switch places with this big R on this chromosome, and then you'll have a Y, a capital Y, with a lowercase r on the same chromosome, and that's the chromosome that gets put into the gamete. Okay, the next one, we could keep them the same, like no recombination happens, and the little y and the little r get passed on, right? That's no recombination. And then same thing for the other one, right? It could just get passed on a capital Y and a capital R. Capital Y and a capital R. And then finally, we could get this lowercase, or we could get this uppercase R with the lowercase y. So there could be recombination here, where the lowercase y switches place with the uppercase y, and now you have a lowercase y on the same chromosome as the uppercase R. So lowercase y, uppercase R. All right, now that we figured out all the possible gamete combinations that heterozygous individual can make, we need to figure out all the possible gamete combinations that this individual can make. Now, I've already done part of the work for you because I've told you there's only one possible combination, right? So let's say that these 2 switch places. The lowercase y switches place with the lowercase y. Okay, what's the big deal? Now you still have lowercase y and a lowercase r on the same chromosome. What if the r switch place? There is no way that you could order these alleles that would be anything other than a lowercase y and a lowercase r on the same chromosome. Even with crossing over and everything, there's no way to reorder these in any other way other than just a lowercase y and a lowercase r. And now all we have to do is fill in our matrix and determine how many of these individuals are both yellow and round. So this first one, capital Y from dad, we'll call this top one dad, lowercase y from mom, and then 2 lowercase r's. that individual is yellow, but they're wrinkled. Next one, 2 lowercase y's, 2 lowercase r's. That one is not yellow and it's not round, so it's green and wrinkled, so not that one. Then we have a capital Y, a lowercase y, a capital R, and a lowercase r. Okay, so this individual is yellow and round. And then finally the last one, 2 lowercase y's, an uppercase r, and a lowercase r. That individual is green but round, or it's round but it's green. So we're asking how many, if these individuals, like if we made offspring, right? We made 100 of these. How many of those 100 individuals do we expect to be yellow and round? Well, we know 1 out of every 4 is going to be yellow and round. So that's 25% of 100 are going to be yellow and round. So what's that? 25. That easy. All right, so we got hemophilia. Hemophilia is a recessive disease carried on the X chromosome. So it looks like this. X, and it's recessive, so that's hemophilia. So that's hema, and then X with a capital H is normal. So we kind of get that straightened out, right? All right, so if we have a carrier mother who has offspring with an unaffected male, what percentage of her male offspring would have hemophilia? Let's figure this out. So a carrier mother, we'll put her up here at the top. So a carrier mother is going to have an X chromosome that's normal, that codes for normal platelets. And then the other X chromosome, since she's a carrier, has that recessive disorder on it, right? The hemophilia. Now, she's okay because she can make platelets. She has a good copy of the gene. She has offspring with an unaffected male. So his chromosomes are XY. Because he's unaffected, we know he has to have a good copy of his X chromosome. He's got a good copy of platelets. If he was affected, he would have the lowercase h. So he's unaffected. Well, let's check this out. So all of the daughters will have XX, right? So one of the daughters is not going to be a carrier at all. She's just going to have totally normal platelets, and none of her offspring down the line will ever be affected by hemophilia. But she might have a sister who gets the good copy from dad and gets the bad copy from mom, and so she's a heterozygote, but she's unaffected because she still has that good copy, and so she's not going to be affected, but she will be a carrier. Now let's go down to the males. So one of her sons is going to receive her good copy of her X chromosome, and he's totally fine. He can make platelets. The other son, however, is going to receive the bad copy of the X, and he's got no other X chromosome to protect him, and so he is going to express hemophilia. Now pay attention here to the question. What percentage of her male, not her total offspring, just her male offspring would have hemophilia? Well, there's 2 different ways to make males. One is with her good X and one's with her bad X. So it looks like 50% of her male, 1 out of every 2 males, will have hemophilia. So percent of males with hemophilia would be 50%. All right. Okay, so we're starting off with grandma's colorblind. So women are circles, and grandpa is not colorblind. They have 4 kids. 1, 2, 3, 4. 2 boys, 2 girls. So here's their 2 girls, and there's their 2 boys. The girls are both carriers, so I'm going to half shade their circles. And both boys are colorblind, and we know that has to be the case because mom's colorblind, and she only has bad copies of her X. And since they get Y from dad, they're only going to get a bad copy from mom. So both boys are affected. Okay. The two girls married 2 unaffected men. So here's your 2 unaffected men they married. Where are we? Boys, unaffected men have 2 children each, a boy and a girl. So there's your girl, and there's your boy, your girl, and there's your boy. But the two colorblind boys, that's these two, marry two normal women and have 2 children each. So there's your spouses. So 2 normal women, a boy and a girl. So they have 2 children. They have a girl and a boy. A girl and a boy. Okay? You can fill in the grandchildren's pedigree designation however you like, as long as they obey the rules of inheritance. Okay, so this should be good, right? So, here's what we're going to do. Get rid of that. Okay. So here's our pedigree. It said you can fill in the grandchildren's however you like, as long as they obey the rules of inheritance. So the male is always going to pass on. If the male is affected, that means he has a bad X chromosome, and so he's always going to pass that on to his daughters. So the daughters are going to be carriers. So I shaded them. I shaded them in half for these guys. Notice the males, I didn't put anything there because they're getting the Y from dad, and their moms we're going to assume are not carriers of colorblindness since they come from outside the family. Now the daughters over here, we can say, okay, maybe one of their sons happens to get one of their bad X chromosomes, so one of their sons is affected. We'll say that maybe one of the daughters happens to get the bad copy of the X from mom, and so she's a carrier. And remember, the dads are from outside the family. And so that's how I'm choosing to complete this. You could complete it differently. You could do both sons are affected. Normally the daughters would not be affected if these affected males were married outside the family. Normally the daughters would just be carriers. But yeah, you can fill in the other male, or you could fill in this other female as half a circle. So that's that pedigree, something like that. Okay, now we get to consider the following pedigree. So consider this pedigree down here, and then answer these questions. So is this dominant or recessive? Well, let's take a look at the pedigree. We have somebody that's affected, right? And he has 3 children, and none of them are affected. So it's possible this individual is a heterozygote, and he passed on his recessive allele to all these kids, and that it's a dominant disease. That's still possible, right? One way that we can tell whether it's recessive or dominant is if we find any affected individual that has 2 parents that are not affected. So look around. Let's see. This individual has an affected parent. This individual came from outside. They're affected. They're obviously affected. This individual right here, though, she comes from 2 parents that are not affected. So this is a recessive disease. Okay? An affected individual comes from 2 unaffected parents. That's how we know. That's my explanation. Is it autosomal or X-linked? Well, there's a couple ways to figure this out, right? If it's X-linked, then we know that the sons of all affected mothers will have to be affected. So let's see if that's true. Let's start at the bottom. So let's find the boys. So here's a boy, and he has an affected mom. So far it looks X-linked. Here's a boy. Here's his mom. She's affected. So far it looks X-linked. Come over here. Here's a boy. Here's his mom. Now, are there any boys who have a mother? Are there any unaffected boys that have an affected mother? And I see one here. See this? That individual is an affected mom. All of her sons should be affected, but only one of them is. So I'd say this is not X-linked. It's autosomal. Because an affected mother has an unaffected son. An affected mother has an unaffected son. All right, let's figure out the genotypes. Okay, so it's a recessive disease. It's autosomal. So let's figure out the genotype of number 8. So here's number 8 right there. Okay. All right, let's take a look. We know we can do this a couple different ways. So we know that this individual, since this is a recessive autosomal disease, this individual is recessive. Right? We don't know what this individual is, but we assume that they do have a dominant gene that's keeping them from being affected. So it's kind of like what is their genotype, right? And we're trying to figure out this individual's genotype. So there's his parents, or there's her parents, I should say. So there's her parents. But let's take a look at her offspring. So it's a recessive disease. So we know that she is unaffected. She has a capital B. She obviously got that from mom. And she probably got this lowercase b from dad. And the reason we know that is because if you take a look, her and her husband had an affected daughter. And so that means both her and her husband have to be at least carriers of the disease. So they're both big B, little b. And that this individual just happens to be little b, little b. They got the two little b's from each, one from each parent. So we assume, we actually still don't know what this individual's genotype is, because we know that this individual is a carrier. So genotype of 8 is big B, little b, and we know that because they have an affected child, but they are unaffected. So they have an affected child, but they are unaffected. Okay, genotype of number 9. Well, we already did that one, right? If they have an affected child, we know that this individual also has to be a carrier, so big B, little b. Right? Or you could also, like, you don't have to use B, you could just say heterozygous, heterozygous if you wanted to. Okay, and then we say they have affected child, but they are unaffected. That's our evidence. They have an affected child, but they're unaffected. And then genotype of number 6, that's this male right here. And so let's take a look. So we know this individual, number 6, is going to receive a little b from his dad. We know that genotype. Now, mom could have passed on, if she was heterozygous, she would have passed on her little b and he would be affected. So she had to have passed on her big B. So number 6 is big B, little b. And we know that because dad's affected, and then number 6 is unaffected, and then number 2 passed on the capital B. So that's how we know. We know that the dad over here was affected, so he obviously passed on the recessive disease to his child. The child is unaffected, so he had to have gotten at least a dominant allele from his mom, right? And let's see. And so he's unaffected. His dad's affected, he's unaffected, and his mom must have passed on. Because he's unaffected, his mom must have given him a good copy of the gene. A good copy of the gene, a good allele. And so that answers the questions for this worksheet. I hope you enjoyed it. I'll see you guys later.